Hello to you all. My name's Jasmine and I'm fortunate enough to be the 2025 Ted Taylor Awardee. Thank you for inviting me to speak at this seminar, and thank you to anybody who read my thesis. I hope you found it somewhat interesting. A little about what I'm currently doing. So I've just started a postgraduate research degree with Professor Cassis at the University of New South Wales, where I'll be building upon the work conducted during my honours thesis. So today I'll be sharing with you all what I did during my honours year, which was my first real introduction to taxonomy and systematics. So Earth's biodiversity is responsible for many essential ecosystem functions, yet is rapidly decreasing, driven by factors such as climate change and habitat destruction. To help manage and conserve our natural world, we need to document and describe unknown species, and there is increasing pressure for this to occur, with biodiversity loss and the threat of the current extinction crisis. So taxonomy is the study, description, naming and classification of Earth's biodiversity, and is closely integrated with systematics, which investigates evolutionary relationships between organisms, allowing for robust species classifications. By documenting species and taxon relationships, taxonomists and systematists contribute to a wide range of sectors, including human health, agriculture, environmental protection, and natural resource management. However, despite the importance of such work, much of Earth's biodiversity remains unknown to science, as we can see here, where despite having over 2 million species described worldwide, it's estimated that around 80% of species still remain undescribed. Australia, in particular, has high endemism and species richness due to a long period of geographic isolation, ranking among the 17 most biodiversity-rich countries and being classified as a megadiverse country. So these megadiverse countries occupy about 10% of the world's land surface area, whilst providing habitat for over 70% of Earth's terrestrial biodiversity. Despite Australia's significance in maintaining Earth's biodiversity and ecosystem functions, only an estimated 30% of Australia's biodiversity has been described. So this knowledge gap exists despite the relevance of having thorough taxonomic and systematic knowledge. Factors such as a decrease in taxonomic specialists over time and unevenly distributed taxonomic focus across taxa are some of the reasons that are responsible for this impediment. Now, insects are incredibly diverse, as we can see from this slide, which has just a small selection of the insects I've spotted around Sydney. So they represent the largest proportion of the gap in our taxonomic knowledge, with an estimated 5.5 million of 8 million species yet to be described. Yet, these taxa are often overlooked, despite having key roles in producing and regulating ecosystem services, including pollination, biological control, decomposition, and nutrient flow. So the hyperdiverse terrestrial heteropteran bugs of the family Miridae are a case in point, exhibiting a significant taxonomic impediment. This group, commonly termed plant bugs, includes economically significant plant pests, as well as others that have been identified as biocontrol agents. Much of the Miridae have been observed to be phytophagous, meaning plant feeding, with roughly 60% of mirid species being host-specific, which some hypothesize may be the result of coevolution between mirids and their host plants. Overall, despite the Miridae being one of the most species-rich insect groups with over 12,000 described species across the globe, it's estimated that over 50% of mirid species remain undescribed. In Australia, they're greatly understudied compared to other biogeographic regions, such as the Palearctic, for example. The cumulative number of mirid species described over time are depicted in this graph, differentiated by biogeographic regions, as we can see along the right-hand side. And yes, although this graph ends in 2009 and we're currently in the year 2026, we can see a significant gap between the number of described species in Australia, which is significantly lower than what one would expect in comparison to the rest of the world, particularly as we can see regions such as the Palearctic and the Nearctic starting to plateau. Thankfully, this value has since increased with programs such as the Plant Bug Inventory and Bush Blitz expanding collections across Australia, and this has been accompanied by increased taxonomic activity with the descriptions of hundreds of new species. The taxon diversity of Australian mirids remained largely unknown until the Zoological Catalogue of Australia was published in 1995, providing a database of all the known Australian Heteroptera. At the time of publication, the Heteroptera totaled 55 families, 779 genera, and 2,006 species. And focusing on one of these families within this catalogue, 6 genera and 9 species were listed as part of the tribe Austromiris, including the type genus Austromiris, which was the focus of my honours thesis. So to give some background on this understudied endemic Australian genus, only 2 species have been described, with Kirkaldy first describing the genus and its type species, Austromiris viridissimus, in 1902, and Poppius describing the second in 1911. This second species was originally placed in its own genus, with the type species named Protomiris nigronasutus. This was later synonymized with Austromiris in 1995. In the original descriptions of Austromiris viridissimus and the then Protomiris, now Austromiris nigronasutus, these initial descriptions were mainly based on features such as colour and the length of some of the structures relative to each other. For example, let's take a quick look given for the description of Austromiris viridissimus shown here on the left. So all of these sections highlighted here focus on the coloration of this species, mainly as seen on the dorsal side of the insect. And this description of the coloration is accompanied by this section on the general lengths and proportions of various structures, including the antennal segments, the pronotum, and the rostrum in relation to the coccyx. And there's a very similar situation happening in the description of Austromiris nigronasutus on the right-hand side, although you may just have to take my word, well, not my word, Google Translate's word on that one. But altogether, the genus Austromiris as a whole was characterized by traits including the following: an apically rounded head, a pronotum with an almost medial constriction as shown by the arrow there, prominent posterior lateral angles, and a sinuously emarginate posterior margin marked in the red dotted line there, which leaves the apical region of the scutellum exposed. Additionally, in 2008, Cassis further observed that the distinct process that is the frons protrudes in such a way that the clypeus is either not visible or barely visible when viewed from above. However, these descriptions lacked any examination of male genitalic structures, which are now known to be essential for differentiating taxa in the Orthotolini, which is the subfamily that Austromiris belongs to. Now, before I go on and continue to talk about the work that was completed during my honours thesis, I want to outline a few minor hiccups that I've encountered over the past few months, and consequently the corresponding changes I've made to this work since I submitted the original version to the Society last year. There are a few changes in the composition and position of species in the phylogenies, because unfortunately, as it turns out, a big challenge to present-day me are actually decisions past me made, but that's unfortunate. So the changes are as follows. So Austromiris hildeber is now replacing Austromiris arida. So originally I started extracting morphological data from Austromiris arida. Austromiris hildeber had already been sequenced, although it hadn't been identified or named yet. So it was sequenced as Austromiris sp. So I dissected the specimen and misidentified it as Austromiris arida, unfortunately, and combined these two data sets, which is not correct.
After this mistake was realised, I later went and extracted morphological data from Austromiris Kirkaldy, as Austromiris arida had not been sequenced, and combined the correct data sets, so now it all belongs only to Austromiris Kirkaldy. Additionally, I was recently able to examine and photograph the type specimen of Austromiris nigronasutus. So this is the original specimen that the species description by Poppius was based upon. So the specimens I completed this original work on had been incorrectly identified as this species based on the original description by Poppius. However, as I mentioned earlier, these descriptions were mainly based on coloration and other external characteristics, which can be a little bit broad at times. So what this meant was that this species, on the left here, was originally called Austromiris nigronasutus, and this supposedly new species was named Austromiris fluorescens. So luckily the type specimen is a male and had already been dissected. So I was able to compare this dissection to the of the type to that of the specimens I'd been working on. So what that meant was that the real Austromiris nigronasutus was actually the specimen on the right hand side here, and this was given the correct name. And the imposters on the left hand side were assigned the name Austromiris fluorescens instead. And finally, there were originally 45 morphological characters used. These have since been refined and reduced to 42. So that's partially due to some of the previous changes that were made. So today, I'll be presenting the most updated results to you all, incorporating all of these fixes. So to my knowledge, it's the most accurate information on Austromiris. And thankfully, the overall conclusions of my work remain the same. So let's talk about that now. I went into this project with three main aims. So firstly, to determine lineages of taxa that share a common evolutionary ancestor in the Austromiris, to test relationships between species in Austromiris to determine whether it is a monophyletic genus, and finally, to undertake a taxonomic revision of Austromiris and the species assigned to it. So I examined 79 specimens across 12 different species. In addition to the NSW collection, the Austromiris specimens were loaned to us from the various institutions listed here and were collected across almost all Australian states and territories. Of the 12 species I looked at, 3 of these were outgroup taxa. These were Nigronasutus bicolorata, Latinova jacki, and Sinostropa elongatus. So these were selected as a reference group to test whether Austromiris was monophyletic, with Latinova and Sinostropa both currently classified in the same tribe as Austromiris, which is the Austromiris, whilst Nigronasutus belongs to a different tribe, the Orthotolini. The remaining 9 species all belong to Austromiris, with 2 of these already having been described, and the remaining 7 provisionally assigned to this genus. Internal and external morphological structures were examined through male genitalic dissections, as we can see on the left-hand side, scanning electron microscopy as viewed on the right, and morphometric character measurements, resulting in 42 characters for differentiating these species. Now, please bear with me, we're going to make a short detour in this presentation, and I'm going to quickly explain some of the terms I'll be using today for these genital structures, with Austromiris viridissimus as an example. So here on the left is an illustration of the dissected male genitalia drawn by the fantastic Hannah Matthews, and down at the bottom is an SEM image of an intact non-dissected specimen. So this final segment of the abdomen, this is a lateral and this is the ventral view, is actually this structure and all of these structures within. So we'll start with this structure here, which is named the pygophore. So this is the genital capsule, which contains the rest of the structures, which we'll go through now. So next up are these paired appendages, which are the left and the right parameres, which sit within the pygophore, and they protrude out of the left side and the right-hand side. And these are asymmetrical structures used as claspers when mating. So this structure here is the Phylotheca, which is positioned between the two parameres and protrudes out the top here. And it contains the next structure, which is the adiagus, the male reproductive organ. We'll go into some more detail with the adiagus, which has the dorsal endosomal spicule, or DES. So here it's referred to as the first dorsal endosomal spicule, as the number of these can vary. Additionally, whenever the dorsal endosomal spicule is present, this section of the dorsal endosomal spicule can be referred to as the keel, which attaches to the inside of the Phylotheca. And the final structure I want to focus on is this one here, which is named the proximal endosomal spicule, which describes its relative position to this structure here, which is the secondary gonopore. So with all of these fun structures in mind, here's the matrix of morphological characters I was able to extract from these 12 species. So we can see amongst all 42 characters, 34 of these involve genital structures, which really reinforces the importance of these differentiating taxa in the Orthotolini. Moving on to the molecular sequences. These were curated from unpublished data in the Cassis laboratory, so part of Dr. Marina Cheng's PhD thesis, and involved an examination of CO1, 16S, 18S, and 28S molecular sequence alignments. And altogether, 3 main phylogenetic analyses were performed using only morphological data, only molecular data, and finally a total evidence analysis combining both morphological and molecular information. So let's take a look at the results. This tree here is based only on morphological data, using maximum parsimony, which selects for the tree with the minimum number of changes in character states, so think Occam's razor. Both an equal weights analysis and an implied weights analysis were conducted on the morphological data, and these both produce phylogenies with identical topologies, which means that the same positions and relationships are shown between the species present here. So shown here is only the implied weights analysis, with Austromiris shown as monophyletic with all of its assigned species in this clade, separate from the outgroups. You can see here that there is strong support for this, given a symmetric resampling support value of 90, and with 6 synapomorphies, which are shared derived characters, uniting these 9 Austromiris species. There are 2 subclades seen here and here, and note the position of the 2 described species, Austromiris nigronasutus and Austromiris viridissimus. And just hang on to this, we'll keep this in the back of your mind, and we'll come back to this later. 2 different methods were used to produce the 2 molecular phylogenies, one using maximum likelihood as seen on the right hand side, and the other using maximum parsimony analysis. So this first tree is the unpublished molecular RAXML tree, and this was produced by Dr. Marina Cheng a few years ago as part of a much larger phylogeny. And the Austromiris species and the outgroups were extracted from it to produce this tree here. So we can still see the same 4 species that formed subclade 1 in the morphological phylogeny being grouped together with significant support, as we can actually see here, and most of the species from subclade 2 are grouped together above without significant support. So there's a polytomy present at this node here, as these 3 taxa groups branch off of here. So this could mean that we're missing data, which would show how these descendant lineages are related, and this could be due to the absence of molecular data for the type species Austromiris viridissimus, which, as you may recall, was grouped with these 4 species in the morphological phylogeny. Additionally, the presence of Sinostropa elongatus amongst these Austromiris species further suggests that the current species assigned to this genus may be non-monophyletic. And using the same unpublished sequence data, this parsimony molecular tree was produced, and the species assigned to Austromiris have again been grouped as monophyletic clade, but as we saw similarly in the previous molecular phylogeny, the grouping also lacks significant support. So the same species that were grouped together to form subclade 1 in the morphological phylogeny are again clustered together here. And some of the species from subclade 2 appear at the top here, although they are paraphyletic. However, it's important to note that there is still no molecular data available for the type species Austromiris viridissimus, which sat amongst these species at the top here in the morphological phylogeny. And again, this may explain the lack of statistical support for a monophyletic Austromiris. Putting all of the available information together produced this tree, which is the total evidence phylogeny. So the topology of this tree is identical to the morphological only phylogeny, aside from the position of these two species, which are identical to the molecular phylogeny, with Austromiris nigronasutus being the basal taxon instead of Austromiris denticulatus. And again, here are the positions of the described species, and we can see strong support for Austromiris as a monophyletic genus with a support value of 89. So 6 anapomorphies support a monophyletic Austromiris, with all of these species sharing the following: a conical head shape, and anterior process on the fronts, so this shape here, an excavated frontoclapial boundary, a C-shaped right paromere, apical branching on the proximal endosomal spicule, so up the top here and here, and whenever the basal keel of the first dorsal endosomal spicule is present, the keel is significantly shorter than the spicule. However, despite these 6 characters being shared across the 9 species of Austromiris, I've made the taxonomic decision to resurrect Protomiris as its own valid genus and reverse its synonymization with Austromiris. So why might I do this? Across each of the phylogenies, a pattern of 2 diverging subclades has emerged, which shows the 2 described species clearly divided, providing support for this decision. This consistent separation of species, supported by the unique character combinations that we see here, supports Protomiris' resurrection and the separation of species into these 2 genera. So, with these new changes, our phylogeny should now look something like this. So the newly designated Austromiris subclade now includes the type species Austromiris viridissimus, as well as 4 new species. And the genus is now defined by 2 key genitalic characters, which are illustrated here. So firstly, there is this process on the right paromere that originates either more medially or more basally. And there is this proximal endosomal spicule that is either more straight or arcuate in shape. The newly resurrected genus, Protomiris, consists of the type species Protomiris nigronasutus alongside 3 new species and is defined by 3 characters. The first being an ovoid body shape, a left paromere with a recurved C shape, and a medial process on the right paromere that is longer than the apophysis at the top. So, just to sum up the composition of the 2 genera, Austromiris now contains Austromiris viridissimus, Austromiris diminutus, Austromiris hilteba, Austromiris naumani, and Austromiris roseo cuneatus. And the newly restored genus, Protomiris, now contains Protomiris nigronasutus, Protomiris borrobini, Protomiris denticulatus, and Protomiris fluorescens. And overall, this is just a preliminary examination of the genus Austromiris, leading to the redefinition of a monophyletic Austromiris and the resurrection and reinstatement of the junior synonym Protomiris as its own genus. However, more work must still be done, as this is only a small step towards closing the immense taxonomic knowledge gap in Australian mirids. So, what's next? I'll be continuing on this work as a part of my PhD. So I hope to examine female specimens of the species mentioned today, and I will be looking at many of the remaining species that are currently provisionally assigned to Austromiris that were not examined in this work. So, many more dissections to come. I also hope to obtain Austromiris viridissimus specimens, so fresh specimens, for sequencing to better clarify the molecular relationships in Austromiris. So if anyone has any specimens, please send them my way. Additionally, I aim to investigate the host plant associations of these species, as the subfamily Orthotolani, which this genus Austromiris belongs to, have often been found in close association with woody perennial shrubs, but more data on the host plants of these different species still needs to be collected. And furthermore, I'll be extending the subject of my research to the subtribe Austromirina, so some of which are pictured here, where I hope to determine a number of things, including the question of whether or not it is monophyletic. But still early stages, so we'll see where I end up in a few years' time. And finally, I want to acknowledge and give a massive thank you to everyone in the Cassis Insect Laboratory for making my thesis possible. And thank you very much to all of you for letting me share these results with you today. So thank you.